Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. My name is Syahmi bin Rahman and my metric number is 203217. So today I will be explaining and demonstrating the experiments in basic technique of microbiology, which are strict plate method or single isolated colonies, as well as gram staining. The strict plate method is a rapid qualitative isolation method where it is used for isolation of discrete colonies, initially require that the number of organisms in the inoculum to be reduced. Essentially, it is a dilution technique that involves spreading a loopful of culture over the surface of the agar medium. Following inoculation, individual cells will be far apart on the surface of the agar medium. Streaking for single isolated colonies or pure cultures can be obtained by picking well-isolated colonies and restreaking these on fresh agar plates. In the streaking procedure, a sterile loop is used for obtaining an uncontaminated microbial culture. The process is called picking colonies, when it is done from an agar plate with isolated colonies and is transferred to a new agar or gelatin plate using a sterile loop or needle. The inoculating loop or needle is streaked over an agar surface. On the initial region of the streak, many microorganisms are deposited, resulting in confluent growth of microorganisms of the entire surface on the agar medium. The loop is heated again between streaking different sections. This will dilute out the sample that was placed in the initial region of the agar surface. Thus, lesser microorganisms are deposited as streaking progresses. Gram staining. Gram staining is a differential staining procedure that are applied to separate most bacteria into two groups on the basis of cell wall composition: gram-positive bacteria and gram-negative bacteria. The differences in cell wall composition of gram-positive and gram-negative bacteria accounts for the gram staining differences. During gram staining procedure, 4 chemical reagents are applied sequentially to a heat-fixed smear. First is the application of the primary stain, which is crystal violet. Crystal violet will stain all cell purple. Second is the application of mordants using iodine solution. Addition of iodine forms a crystal violet iodine complex, which intensifies the color of the primary stain. Next step is decolorization, which distinguishes gram-positive and gram-negative bacteria. Application of decolorizing agent such as ethyl alcohol or acetone may or may not remove the primary stain from the entire cell or only from certain cell structures. Gram-negative bacteria have much less peptidoglycan in their cell walls, so this step essentially renders them colorless, while only some of the color is removed from gram-positive cells, which have more peptidoglycan and much thicker cell wall. The thick cell wall of gram-positive cells is dehydrated by the decolorizing step, causing them to shrink and trapping the stain iodine complex inside. After the decolorizing step, a counterstain is applied, which is usually safranin, to color the bacteria pink. The decolorized gram-negative bacteria are ready to pick up the pink stain. Gram-positive also pick up the pink stain, but it is not visible over the darker purple of the gram-positive bacteria. If the staining procedure is performed correctly, gram-positive bacteria will be purple, while gram-negative bacteria will be pink.